In this video we will discuss about the topic where we will understand how this TAGLN2 helps in T cell metabolic fitness. So what this TAGLN2 expression on the cell surface of the T cells does is that it will promote lipid uptake, and that will help in maintaining the metabolic process of the CD8 plus T cells. In the previous video we have understand that TAGLN2 expressions goes down whenever there is a ER stress in the T cells. So in this video we will discuss if these TAGLN2s are expressed on the cell surface, then it will helps in the uptake of lipid metabolism, and that will helps in maintaining this metabolic fitness of the CD8 plus T cells. So in the panel A they have shown that with increase in ER stress there is a decrease in the FABP expressions on the cell surface. This is similar to what we have discussed in the previous results or in the previous video. So this things, the thepsigargin is used to induce ER stress, and we can see that with increase in the thepsigargin concentrations there is a decrease in the FAB expressions, and these things are quantified on the panel B also, where surface FAB expressions are goes down whenever there is a ER stress which is induced by TM. And next they check the lipid uptake of this ER stress induced cells, and what they have find out is that lipid uptake goes down with increase in the ER stress, which is measured by done by this TM reagent that cause ER stress, and these things are quantified on the right hand side with the dose dependent TM reagent leads to the decrease in the uptake of the lipid. And they did the same study with other two reagent also, that is 2DG and TG, and all the three reagents shows the similar results. So this evidence, this result shows that with increase in the ER stress there is a decrease in the FABP expressions, and that will cause decrease in the lipid uptake. However, when they use the IRE1 alpha knockdown or XPP1 knockdown cells, they see some of the reverse results, and reason we have discussed in the previous video. So in the case where IRE1 alpha and XPP1 knockdown occurs, we can see that with increase in the thepsigargin or reagent we see the increase in the FABP expressions. It can be seen in the panel C in the IRE1 alpha knockdown cells, that is surface expression of FABP goes up, and same things they have seen in the XPP1 knockdown cells also. With increase in the thepsigargin or with increase in the ER stress reagent, they see the increase in the FABP expressions in the knockdown cells compared to the wild type, as this XPP1 and IRE1 alpha will trigger the downregulation of this FABP, and when those two proteins are absent or knocked down, they will not be able to downregulate FABP5 proteins on the cell surface. And thereafter what they did is they check a lipid content in those knockdown cells, and they see that lipid uptake is high in the knockdown cells, both in the case of IRE1 alpha and also in the case of XCP1 alpha. We can see that in the knockdown the lipid uptake is high. So this indicates that the lipid uptake directly related to the FABP5 expressions on the cell surface. And thereafter they check the dose dependent oxygen utilizations of this of the cells, CD8 plus T cells, under different concentration of this TM. And what they have find out is that with increase in concentration the oxygen's utilization goes down using the OCR experiment, and these things are quantified on the right hand side of the panel E, where we can see that basal respirations and maximal respirations goes down with increase in the ER stress because of this TM reagent. And thereafter what they did is they overexpress the TAGLN2 expressions in the ER stress cells and wanted to see whether it will have any negative impact on the lipid uptake or not, not negative, positive impact on the lipid uptake or not what they have find out is that when the cells in which they use TAGLN2 overexpression, they see the increase in the lipid uptake. Same things they have seen, same things are shown in the panel F, where we can see that where overexpression of the TAGLN2 will cause more lipid uptake, and this thing they quantified using the OCR measurement or oxygen utilizations by the cells that are under ER stress, and the TAGLN2 mRNA is overexpressed. You can see that when the TAGLN2 mRNA is overexpressed, there is more utilization of oxygens, indicating that the basal respirations and the maximal respirations are high in the conditions where TAGLN2 expressions are high, are overexpressed, or TAGLN2 mRNAs are overexpressed. And the simultaneously uh, other markers are also overexpressed, such as CD44 and Ki67 and this IFN gamma PE, and this CD44. This is the quantifications of this IFN gamma PE, and also this granzyme, that is GZMB, and it is also upregulated, which is necessary for triggering of apoptosis in the tumor cells. So, and we have discussed about these granzymes in one of our previous videos, how this extrinsic apoptosis occurs through granzyme. And once they have validated that with upregulations of these TAGLN2 expressions, they can see the increase in the lipid uptake and some of the other markers. Then what they did is the next time they deactivate this TAGLN2 mRNA using this etomoxir, and they wanted to check whether they see the reverse results or not. And when they did that, and in the case where TAGLN2 mRNA is inhibited, they see a decrease in this oxygen utilization by mitochondria. And same things are shown on the right hand side of the panel K, where we can see that where the inhibitor is used, the OCR levels goes down, or of oxygen mitochondrial oxygen utilizations goes down. And also we can see that the CD44 levels and Ki67 levels levels also goes up in the CD8 plus T cells, where this OA is used. So this indicates that the effect of this TAGLN2 is specific to the uh, effect of this TAGLN2 expressions are specific to this lipid uptake, but it does not affect the other factors such as CD44 and Ki67, as they are still goes up, similar to the case where TAGLN2 expressions are overexpressed. And after validating that, they wanted to check this effect of OA and under different conditions. And what they have find out is that in the case of TAGLN2 knockdown conditions, and TAGLN2 wild type conditions, and the complete conditions, the glucose free conditions, they can see that when they inject this OA, and they are able to see increase in this lipid uptake. And these things are quantified on the right hand side of the panel M, where we can see that when OA is not introduced, they see the decrease in the lipid uptake activity, whereas when OA is introduced, they see that the restoration of this lipid uptake activity. So this results indicates that is a TAGLN2 expressions that helps in the T cell metabolic process through lipid uptake, increase in the lipid uptake. If you want to understand more about these results in detail, you can go through the reference. that has been provided in the description. That's all for this video. Thank you for your time.